Welcome to this lesson on DNA structure. DNA, or deoxyribonucleic acid, is one of the most important molecules in all living organisms. Today, we'll explore its famous double helix structure, often described as a twisted ladder. This elegant shape holds the secrets to how genetic information is stored and passed from one generation to the next. DNA stands for deoxyribonucleic acid. It is the molecule that carries all the genetic instructions needed for the development, functioning, and reproduction of every living organism on Earth. Think of DNA as a biological instruction manual that tells cells how to build proteins, when to divide, and how to carry out their specific functions. Without DNA, life as we know it would not exist. The structure of DNA was discovered in 1953 by two scientists, James Watson and Francis Crick. However, their discovery was built on crucial experimental work done by Rosalind Franklin and Morris Wilkins. Franklin's X-ray crystallography images, particularly Photo 51, provided key evidence that DNA had a helical structure. This groundbreaking discovery earned Watson, Crick, and Wilkins the Nobel Prize in 1962. DNA is made up of two long strands that twist around each other, forming what we call a double helix. Imagine a ladder that has been twisted along its length. The two sides of the ladder represent the backbone of the DNA molecule, while the rungs connecting them are formed by pairs of chemical bases. This twisted shape is not just beautiful, it's also incredibly functional for storing and protecting genetic information. DNA has two main structural components. First, there's the sugar-phosphate backbone, which forms the outer framework or sides of the ladder. This backbone is made of alternating sugar molecules called deoxyribose and phosphate groups, providing stability to the entire structure. Second, we have the nitrogenous bases, which form the rungs or steps of the ladder. These bases connect the two strands together and carry the actual genetic code. There are 4 nitrogenous bases in DNA, and they fall into 2 categories. The purines are larger molecules with a double-ring structure, and include adenine, abbreviated as A, and guanine, abbreviated as G. The pyrimidines are smaller molecules with a single-ring structure, and include cytosine, abbreviated as C, and thymine, abbreviated as T. These 4 bases are the letters of the genetic alphabet, and their specific sequence along the DNA strand encodes all genetic information. The nitrogenous bases don't pair randomly. They follow strict pairing rules discovered by Erwin Chargaff. Adenine always pairs with thymine, and guanine always pairs with cytosine. These pairs are held together by hydrogen bonds, which are relatively weak bonds that can be easily broken when needed, such as during DNA replication. This complementary base pairing is essential because it allows DNA to copy itself accurately and ensures that genetic information is preserved. The double helix has several important structural features. First, the two strands run in opposite directions, which we call antiparallel orientation. One strand runs from the 5' end to the 3' end, while the other runs from 3' to 5'. Second, the helix completes one full turn every 10 base pairs, creating a regular, repeating pattern. Finally, this entire structure is perfectly designed for 2 critical functions: accurate replication during cell division and stable long-term storage of genetic information.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.